Hello, my name is khaireddinearb. This is my project, AI-powered DNA variant effect prediction platform. I will just go straight to it. This is the website. So I attached this in the attachments while I was submitting this project. So we can just sign up, create an account if you don't have one. And then I'll just show you my account right now as we go. So we just start a simple analysis and I'll just explain every step as we go. We can name it session, or we can name it anything we want. We'll create this new session, wait a little bit till it loads. And here is the platform. It goes on 3 panels. If you're not familiar with it, and if you're watching it and you want to try it, you can simply just click on guide and it takes you into a tour on all the features of the app. So since I'm presenting now, just explain every section and what it does. On the left, we have 2 sections here. We have the genome assembly, and we go here, we select anything we want. Okay, so far we support 6. On we go, we're going to support more. So far we have probably go with the SG38. Here we have 2 things. We have the chromosomes, and here we have the genes. For the chromosomes, if we click on any one of them, we should get the genes inside every single chromosome. Okay, here we have like 3. For the sake of this demo, let's go for click on BRCA. So BRCA, I will show it to you now, is a type 1 cancer, okay? And I included here on the middle a section. And let's just start from the left and go all the way to the right. So as we said before, once we click on it, we can just get all the genes, okay? And assuming we're interested in this one, we can just click on it, okay? Once we click on it, we get like a 3D view of the whole protein here, okay? Here, this is a breast cancer type 1. And it's a susceptibility protein, okay? If we want, we can go even full screen and then check all the structures here. We can even select on any place and then zoom out. It blurs everything on the background, and we can get all the molecules and how the structure of this protein actually exists. Okay, there are a lot of parameters here. If we want to include more things, so you can play with them a little bit. And for the sake of this demo, I will just stop here, and I will just leave you a little excitement to actually try this feature. And here we use the mole star, okay? The mole star is you can think about it as a 3D engine for rendering 3D proteins, okay? So here the sections on the middle gives us this the gene context, okay? It gives us the name, chromosome, the nucleotide, the start and the end, the strand, is it forward or backwards, and the length. And here it gives us the organism things, the scientific name, the common name. It gives us like all the details we need, okay? There are more features that will include as we just proved that the prototype is actually worth working on and it's actually being used by clinicians and people who study basically in the biology sector. Here at the bottom here, we have the sequence viewer, okay? Now the sequence viewer, basically what it does, it loads all the DNA sequence, okay? This DNA sequence here is just full of nucleotides, okay? And here, basically on gene editing, what we do, we sometimes delete, or we just basically change, which we call the substitution, or we just do other mutation types, okay? So let's assume that we're interested in this one. We want to study this nucleotide here, this position here. So once we hover, I think you can keep an eye here. Once we hover, it actually just changes to reflect where we hover exactly. So assuming we're interested in this one, we can just click on it. Here on the right, we have 2 things. First one is a single variant, okay? You can think about it as a single edit. So we go here, tweak some parameters, and then we see like what's going to happen at the end by clicking on the analyze. Here is another feature that I'm working on right now, which is the batch analysis. So sometimes we want to analyze not one by one, but actually want to do like a batch, like a whole sequence of DNA. So here we have the gene and we have the chromosomes, okay? We're now working on chromosome 3, and we are working on this BRCA1 associated long name here, which is the gene inside this chromosome here. So once we create on the nucleotide, we can basically come here and select the mutation type, okay? So the substitution, you can think about it as changing A with TCG, okay? And then we have deletion, it's still experimental, okay? And then insertion, which is also experimental. So once we prove that this one is being used on this substitution, we can actually work and extend our API to just include more mutations. We have more than 6 that we're planning to actually add for our model here. So here it gives us the position, okay, within the DNA sequence, the whole thing. Here there is the reference nucleotide, which is this one that we just clicked on. In here we can just change it with something else. Okay, usually here we just change it with C, G, or T. Okay, there are 4 nucleotides here. So assuming we want to change it with C, we can just create on or click on analyze, and here, since I have an old account a little bit, so it's gonna go here and then do like an analysis here. Okay, so we wait, and usually the inference is gonna take a little bit of time. Let's just show you how the inference actually works. So the model is actually deployed on model.com, and let's just go to the dashboard and see like how the endpoint is actually being sent here. So as you see here, we have one container working right now. It's a status which is pending. You can click on it and view the logs. We're waiting a little bit. So apparently here we got a response, success. We go back and we usually have to see it here. Okay. So what it does here, it gives us something called likely benign. It gives us the delta likelihood score, and it gives us the confidence percentage. Okay. And here is the most important part. Okay, is it likely benign or likely pathogenic? So usually in genes, we have like 5 different categories, okay? And we have something we call the VUS. This VUS is something very, probably you can just view it here. I will try to fetch an example. So for example, this one here shows us that it's uncertain significance. So what we're trying to do is to help scientists basically classify it as likely benign or likely pathogenic. Pathogenic means it's likely to hurt this creature that you're trying to edit this DNA sequence. Likely benign is not really harmful. Okay, you can proceed with this procedure here as you go. So this is one, and this is another one here. Okay, this is known variants from the ClinVar data. Okay, usually we have the ClinVar analysis. It gives us uncertain significance. And here we're going to use our model to just click on analyze, and it's going to go there and give us the analysis and what it thinks. Here, the model here gave us that it's likely benign. Okay, sometimes we have some errors here that the confidence is 100% and delta score is like almost 0, but we're going to work on it as we go. Okay, this is the prototype so far. Probably you're going to see like there are some errors here and there as we just view right now. Okay, here mainly it's working 100%, give us a good confidence, good delta score. Okay, and it gives us a good classifications. Now as we go and prove that this prototype is actually worth proceeding, and it's showing some clinical trials, it's helpful for scientists, we can actually proceed to include other features too. So I even included like some docs here, documentations for the API. You can check every single section here, like the API key, the analyze variant section, API reference, the mutation types, authentication, and some code examples if you want to work with the API a little bit. And here you can find some architecture too and error handling. And we can go to the dashboard, and we should get the session here. And you can play with the prototype as you go, and probably just report back to us if you're interested on extending it with other features too, or you see other areas that you want to report to us. Thank you very much.